Hi, welcome again to Consider This, Simplifying the COVID-19 Conversation. I'm Marianne McDowell, and I co-host this series with Dr. Heidi Beininger-Burnett. This week, we're going to be talking about vaccines and immunology of COVID-19 with guests Brian Baker and Jeff Shorey. But last week, I mentioned a couple of therapies that President Trump received, and I thought I would talk a little bit more about those in detail in this little tutorial. But before we get started on that, I want to remind you of the SARS-CoV-2 life cycle. So the virus interacts with ACE2 receptors on the cell surface of epithelial cells in the respiratory tract. This mediates entry of the virus into the cell where it resides in a sac or a vacuole. That vacuole matures or changes over time. Eventually, the spike protein gets cleaved by host cell enzymes, and that mediates fusion of the vacuole membrane and the virus membrane at releasing the viral RNA into the cytoplasm. At that point, the virus takes advantage of the host cell machinery to read that mRNA or that RNA and make proteins, make viral proteins to eventually create new viruses. Some of those proteins actually read the RNA and replicate it, so make copies of it. They get translocated up to where the proteins are, new viruses get made, and the viruses are released from the cell. So one of the treatments that we talked about last week was monoclonal antibody therapy. So what happens in this case are antibodies actually recognize the virus spike protein and block the interaction of the spike protein with the ACE2 receptor. So the viruses never can gain entry into the cell and start an infection. Two monoclonal therapies that are being experimentally evaluated are Lilly and Regeneron. The Regeneron antibodies is an antibody cocktail of two different monoclonal antibodies, and these are in phase 3 clinical trials. President Trump was able to receive this experimental drug last week. Monoclonal antibody therapy is somewhat similar to therapy with convalescent serum or blood serum isolated from patients that have recovered from COVID-19. But in this particular case, you could have certainly neutralizing antibodies that bind the spike protein, but you could have antibodies that don't neutralize but recognize spike, or antibodies that recognize other components like the nucleoprotein, for example, as well as lots of other proteins and molecules in that particular serum. The other drug that one of the other drugs that President Trump received, it was Remdezavir. So Remdezavir does not neutralize the virus. In fact, the virus interacts with ACE2, gains entry, the vacuole matures, the spike protein is cleaved, fusion occurs, and the mRNA gains entry into the cytoplasm. Even the reading of that RNA and the making of proteins is not blocked. In fact, what happens is the remdesivir actually mimics some nucleotides and just basically blocks the replication of the RNA, leading to truncated RNA molecules. If you're interested in the exact mechanisms, I've posted a YouTube video under additional resources that clarifies how that occurs. So that was two of the treatments that President Trump received. He also received dexamethasone, a steroid that blocks inflammation, which we can talk about later in our live discussion, as well as some vitamins, etc. Well, that's all I have for now. If you have some questions, please email us at consider@nd.edu. I'll see you soon.